So today, we're going to dive into a technology that honestly sounds like it's straight out of science fiction. But what's so wild is that it's actually rooted in one of nature's oldest, most powerful defense systems. We are talking about RNA interference, or RNAi for short, and it's a way we can protect our food by, get this, literally silencing the genes of its greatest enemies.
But look, before we get into how this amazing technology works, you've really got to understand the scale of the problem, the reason we even need it. Because every single day out in fields all over the world, there's a constant, invisible war being fought against this relentless army of microscopic invaders.
Just let that number sink in for a second. Over 30%. That means that almost a third of all the food we could be growing is just gone, wiped out by things like fungi, viruses, and bacteria before it ever gets a chance to get to us. That is a staggering loss, and it has huge, huge implications for feeding everyone on the planet.
For decades, our main answer to this has been chemical pesticides, but we're all becoming more and more aware of the environmental costs, and on top of that, the pests are getting resistant. So the race is on to find something smarter, something safer, something more targeted. So, what's the alternative?
Well, what's so cool is that the answer wasn't some brand new thing we invented from scratch. Nope. It turns out it's a mechanism that life itself has been using to protect itself for millions, maybe billions of years. We just had to figure out how to speak its language.
That's right. Nature is the ultimate innovator. I mean, long before we were even around, plants and animals had already developed this incredibly sophisticated internal security system to fight off invaders. It's nothing short of an evolutionary masterpiece, a way of recognizing and neutralizing threats at the most fundamental level possible, their genetic code.
And the way we found out about it, it's just a fantastic story. It was a total accident back in 1990. Scientists were trying to make petunias more purple, but instead, they ended up with completely white flowers. They had literally turned the gene off by mistake. That happy accident kicked off years of research that finally unraveled the whole mechanism, gave it the name RNAi in 1998, and eventually won the scientists a Nobel Prize in 2006. So, what is it, really? Well, think of RNAi like a cellular surveillance system. When it spots a suspicious piece of genetic material, like from a virus that's trying to invade, it creates a kind of molecular wanted poster. Then it uses that poster to hunt down and shred any matching messages from the invader, basically shutting down its entire operation.
Okay, so let's get into the good stuff. We're going to shrink ourselves down to the molecular level and actually see how this incredible little machine inside our cells works. It is a beautifully choreographed process. The whole thing is this elegant four-step molecular dance. It all starts with a trigger, a piece of double-stranded RNA that the cell doesn't recognize, red flag. That kicks the cell's machinery into action, and it starts to chop up that foreign RNA into little identifiable fragments. Next, those fragments get loaded into a special protein complex, kind of like loading bullets into a magazine. And finally, that complex starts the silence phase, using that little fragment as a perfect guide to hunt down and obliterate any matching enemy genetic code.
Okay, whoa. I know this looks like a super complicated subway map, right? But don't worry about all the lines and arrows. It's basically just showing that whole battlefield we talked about. So instead of getting lost in the diagram, let's just meet the three key players who make it all happen.
First up, meet an enzyme called dicer. Its job is simple but absolutely critical. Dicer acts like a pair of molecular scissors. It sees that long, threatening piece of foreign RNA and just dices it up into these perfect, small, manageable pieces.
Now those little pieces that dicer makes, they're called Small Interfering RNAs, or siRNAs. And you can basically think of an siRNA as a molecular mugshot. It's a short, exact snapshot of the invader's genetic code, and it's about to be passed on to the cell's security team.
And that security team is called RISC, the RNA-Induced Silencing Complex. RISC is the real hero of this story. It's a seek and destroy unit. It grabs our molecular mugshot, the siRNA, loads it up, and then starts patrolling the cell. The moment it finds a message from the invader that matches that mugshot, it slices it in two. Game over for the pathogen.
And this is so cool. You can see it all in action here, up close and personal. Up top, there's dicer, locking onto that foreign RNA to make its precise cuts. And then below, you see the RISC complex, armed and ready with that little guide RNA, finding its target and preparing to neutralize the threat. It's just an elegant and ruthlessly efficient system.
Okay, so here is the really mind-blowing part. Now that we understand this natural process in all this detail, we can hijack it. we can actually co-opt this ancient defense system and turn it into one of the most powerful tools we have for agriculture.
So here's the big idea. Scientists can look at a nasty fungus, right? And they can pinpoint a gene that's absolutely vital for it to survive or infect a plant. Then, in the lab, they can build a piece of double-stranded RNA that is a perfect match for that specific gene. When they introduce that custom-made RNA, it triggers the plant's own natural RNAi machinery to go out and destroy the pathogen's instructions. It's genius.
And there are two main ways to do this. First, there's what's called host-induced gene silencing. This is where you actually genetically engineer the plant to make the specific dsRNA itself, so it has this built-in 24/7 protection system. But the other way, which is really exciting, is a non-GMO approach. You just spray the lab-made dsRNA right onto the plant's leaves, kind of like a regular spray, and the plant absorbs it and puts it to work.
And believe me, this isn't just a one-trick pony. The versatility is what makes it so powerful. I mean, just look at this list. RNAi has been used to protect all sorts of crops against a huge variety of threats. We're talking fungi on wheat and tomatoes, bacteria on rice, and even viruses attacking plum trees. It's a true platform technology.
So when you step back and you put all of this together, it's pretty clear that we could be looking at a full-blown revolution in how we protect our food, moving away from broad-spectrum chemicals and toward these incredibly precise molecular tools.
And here's why this matters so much. First, it's hyper-specific. It's based on a genetic code, so it only targets the bad guy and won't harm helpful insects like bees. Second, the RNA molecules themselves are totally biodegradable. They just break down in the environment. This means we could massively reduce our need for chemical pesticides. And maybe best of all, it's way harder for pests to develop resistance to it.
You know, this quote from the scientific literature really says it all. Scientists see this as safe, eco-friendly, and evergreen. It's not just another tool. It's a sustainable one that lets us work with nature's own playbook to build a better future for farming.
By harnessing a process that's been perfected by evolution over millions of years, we're really opening a door to a whole new world of crop protection. It really makes you wonder, doesn't it? Maybe the future of farming isn't about a bigger tractor or a stronger chemical after all, but about the elegant, powerful simplicity of a single molecule.